All right, so we are going to talk about genetics, and I would like for you to just have this kind of open on one tab and have your notes kind of open next to it. And you're just going to put in your notes your own words. We're going to go through this hopefully quickly. I know vocab can kind of not be the most interesting thing, especially if you're just listening to me talk. So, genetics is the branch of bio that deals with heredity and information and the variation of organisms. Chromosomes are these guys right here. They don't look exactly like this, obviously, but they're what carries our DNA and our information. It is what makes our proteins, and it's what makes us us. Come on, internet. All right, Mr. Technical Difficulties. Anyways, as this is going, our chromosomes appear in pairs. They're called homologous chromosomes. Get one from mom, one from dad. This is why we went over this in meiosis briefly before we split ways. And this is what causes new combinations of genes. It's what we call crossing over. It's where one bit of one chromosome goes to a different bit. You don't need to know a ton of that for genetics, but that's just something to keep in the back of our minds. This happens after fertilization from two parents. This is showing some crossing over here that can happen. And also it's showing the homologous chromosomes. So these genes are kind of the same. So if this is for eye color, this might be brown eyes, this might be blue eyes. So mom's giving you brown eyes, dad's giving you blue eyes. But these guys right here are identical. So brown eyes, blue eyes, blue eyes. Same gene, but not necessarily identical genes. So there's this guy, his name was Gregor Mendel. He was a monk from Austria, and he studied pea plants. He was a very boring guy, and he studied pea plants for years and years and years and realized that he found these traits. So he looked at 7 traits or characteristics of pea plants. He cared if they were short or tall, round or wrinkled, yellow, green, purple or white petals, green or yellow, if the flowers were on the side, axial or terminal, wrinkled or inflated pea plants. So he studied these really closely, cared a lot about it, and we're going to talk about most of our genetics in terms of pea plants. He discovered most of this on accident, but it just so happened to work out well for him because of pea plants having this thing called dominant and recessive. So terms that we need to know, so make sure you have this in your notes. Gene is a single protein. A genome is the entire set of genes. And this is the big one right here. The big takeaway is our allele. So an allele is a version of a gene. So if we have one allele for blue eyes and then an allele for brown eyes, that is a version of a trait. So an allele is just a version of a trait. So you can have the same allele or different alleles. One comes from mom, one comes from dad. So the first thing is homozygous. So zygous is like a zygote. So it's the fetus, it's inside the pregnancy. So homo means same. So homozygous means having identical genes for the characteristic. So you're having the same. Heterozygous means having 2 different. Dominant is the allele that is the one that is seen, and recessive is the one that is not seen. This is going to make so much more sense when we start using letters. So I'm actually going to grab, hopefully, the pen. Give me 1 second to figure out how to do that. I think I might have to exit out of presentation mode Grab a pen. All right. So when we do this, homozygous, we're going to use the letter A. So dominant is always capital. So if I'm talking about, we're going to go back to pea plants. So if I want axial or terminal, or green or yellow, or if I want purple or white, round or wrinkled, whatever we choose, we're going to have a dominant or a recessive. So when I go down here, I'm actually going to delete this, and we're going to do, I'm not going to do A. We're going to do round over wrinkled. I just want to do one that's kind of easy to tell the difference between, and you can tell my different letters. So we're going to do the round and wrinkled. So round is dominant over wrinkled. So round is dominant to wrinkled, right? So round is capital. So if I mate a round plant to a wrinkled plant, it's going to be capital. We're going to see the round pea plant over the wrinkled. We don't use a different letter. We use the same letter when we're talking genetics. Our homozygous, they're going to be the same. So I can be round, or I can be wrinkled. My heterozygous is going to be different, though. Tricky thing here, the really, really, really tricky thing here, though, is what will this look like? Well, based on our theory of dominance, based on what we know it's going to be round because this dominant guy, right? We have a capital R. So anytime we have this big R, it's like it's yelling. So if we have a big R or a big uppercase letter, it's going to be yelling. It's going to say, hey, I'm loud, I'm dominant, I'm going to be the one that's shown. And then we have like this little guy, this little R, he's whispering, he's not going to be shown. You're not going to worry about him. We're going to do lots and lots of practice, so don't worry. I promise it'll make more sense. But there's lots of vocab to go along with this. So then we have our genotype and our phenotype. So genotype is the genes. It's what the genes look like. So that's going to be, this will be like our genotype. And our phenotype, it's our appearance, it's what it looks like. So it would be round. It's really hard to write with clicking of a mouse. I apologize. So our next thing that we need to talk about, this, I'm not going to get too into it. I'm just going to talk about it cursory really quickly because it's here and I want you to know the difference. I'm going to fill it in your vocab sheet because I don't want you to get confused by it. A monohybrid cross, we're only going to do monohybrid crosses to where you cross one trait. And you guys might have done this before with a Punnett square to go across two traits. We're going to go through how to do this step by step. The P generations are the parents. Then you have the F1 and the F2. This is how Mendel kept track. So the first filial and the second filial, it's just the offspring and then the second generation. So if you would look at this as first generation, so this would be like the grandparents, parents, and kids. So Mendel looked, like we said, 7 characteristics. Oops. No. Come back, Google. Mendel looked at 7 characteristics in pea plants. He said that they were either tall or short. They were either inflated or constricted, round or wrinkled, smooth or wrinkled, yellow or green, axial or terminal, purple or white, green or yellow. I have no idea why this picture is so terrible. And there's 0 color. I apologize. A monohybrid cross, we're going to do tall. So our parent generation is going to be tall by short. So these are our homozygous. So we have a homozygous dominant and a homozygous recessive. So we have a big T crossed by little t, little t. So that's our P generation. So in our true breeding, that means that they only have these genes. Our F1 generation, so our parent generation, we cross big T by little t, and we only have big T and little t. That's because the only genes that this guy can give is a big T. When this breaks down, when we split these genes, right? Remember, when we make a sperm and an egg, the chromosomes will split. Remember, it's a reduction division. So they're not in pairs anymore, they're single. So this plant can only give big T genes. This plant can only give little t genes. So our F1 generation is all heterozygous, all big T, little t's. Our F2 generation is going to be a cross between, big T, little t. So we're going to do something called a Punnett square. So we're going to make a square that looks like a window pane. And what we're going to do is we are going to cross our parent generation first. So we're going to figure out how we got that big T, little t. So we're going to put our parents. So one parent goes across the top. Doesn't matter, mom or dad, goes across the top. And one parent goes across the side. So then what we're going to do is we're going to take this and we're going to bring them all the way down, just so you can see where I'm putting them. I'm going to put my big T here, and I'm going to put my big T here. I'm going to do the same thing with this column. Big T here. Capitals always go in front of lowercase. I'm going to go across this way. So this little t is going to go in this box, and this box, this box, and this box. This doesn't make sense at this point. Take the video 30 seconds back, look through it again. If it still doesn't make sense, shoot me an email, call me. We'll talk it out. We'll set up a Zoom, something, because it's just going to get a little more confusing. So then we are going to talk about the genotypes and the phenotypes. The genotypes, remember, are our genes. So these are our genes. So 100% all of our plants are big T, little t. Our phenotypes, what they look like, all tall plants, 100% tall. Now we need to make the F2 generation. This is where it can get a little more confusing. So we have our big T. I'm going to actually erase this and go through it with you guys, just because I want to make sure we've got it. And I hope that you're like, this is so boring, Miss Horton. Why am I listening to you? I really hope so, because that means you've got it under control and you find me boring, and I'm okay with that because that means you understand it. So F2 generation, that is going to be our F1 generations self-fertilize. So that means that they're just going to share pollen with each other. So I put the parents across the top, parent across the side. So again, I put this box here, and this guy goes down and fills with these two. This guy goes down, fills across. Then we go across. Big T, and then little t, because we're across. Big T, little t. Perfect. Okay. So when we get to genotype, we have one big T, two big T, little t's. Then we have one little t's. That means we have 3 tall and 1 short plant. 3-to-1 ratio.
A key to a Punnett square is that we split the parents up. So we have to make the gametes. Remember, gametes are sperm and egg cells. So when we split these up, each gamete goes to a different part of the window pane. It's really, really important that we remember it. Once we have the gametes, we set up the window pane. I'm going to talk through the one on flower color. So purple, big P is dominant to white, little p. We cross big P with little p. And we're going to get this heterozygous all purple flower. We're going to cross our F1 generation. That F1 generation is going to give us these gametes. And again, we're just filling in the Punnett square, guys, just filling it in. So we're going to go back to this idea of Mendel. So Mendel had 3 principles. The first principle that we're going to talk about is principle of dominance. It's that one allele always masked another in the F1 generation. So basically, this is saying that tall will dominate over short, axial will dominate over terminal flowers. This means that whatever we have, purple masks white, tall over short, one is always louder. The next is principles of segregation. This rule right here says that when you have homologous chromosomes, when you have pairs of chromosomes, traits separate. That's all this is saying. So when sex cells are formed, the pairs become separate. So each sperm and egg, each sex cell receives its own type of gene. So this is an inheritance pattern of cystic fibrosis. So this is just a little bit different because this is a recessive gene. So this is just a word problem that you'd have to read a little bit differently. So this is saying if 2 parents carry the recessive gene of cystic fibrosis, so they're both heterozygous, 1 in 4 is considered homozygous for the cystic fibrosis gene and has the disease. Cystic fibrosis is a disease of the lungs that causes the inside of the lungs to make extra thick mucus that can't be removed. And it's very, very difficult for patients with cystic fibrosis to breathe. They oftentimes need a lot of physical therapy to help remove the mucus. They have a shortened lifespan. They have difficulty breathing and moving. But if you are heterozygous, if you're just a carrier, you lead a normal life. However, if you have children with somebody that's also a carrier, 25% of the times you have a chance of having a child with CF. So this is saying that 3 children could be normal and 1 could have cystic fibrosis. Same thing, just flipping it from the 3 tall or the 3 purple, 1 white. You're saying 3 normal, 1 having CF. We're going to skip dihybrid crosses. We are not going to talk about dihybrid crosses at all. Dihybrid crosses are just if you want to talk about 2 traits at the same time, you can do that. We are not going to go into this and we're not going to practice it. The only thing we're going to say is that Mendel had a 3rd law and said that it's called the law of independent assortment or the principle of independent assortment. And that basically means that it doesn't matter if you have a purple or tall, white, short, all of these gametes, all of these genes sort separately. So it doesn't matter what chromosome goes where, it's an equal chance that they will move around. The next vocab word you have is a test cross. So if you have a pea plant, because again, Mendel is a very boring guy, and he just worked with pea plants. So
if you have a organism like a purple flower that is a pea plant, and you don't know if it's going to be homozygous dominant or heterozygous, you have to do a test cross to see what it's going to be. So a test cross is a cross with a homozygous recessive individual. So you know that it has to have at least one capital letter because it's purple, but you don't know what that second one is. It could be an uppercase P, could be dominant, or it could be heterozygous. So the reason you cross it with a white plant is because if you get any white plants, you know that you have a heterozygous plant. So we're going to kind of talk through this test cross. I know that I have one purple flower, but I don't know its second chromosome. I don't know what the second gene is. And if I cross it with, that is supposed to be an X, I'm sorry, a white flower, two little p's. So big P, and if I know that this is guessing that's a big P, put those two there, and I get all purple flowers, that's telling me that all purple flowers means that this must be dominant because it gave me heterozygous flowers. However, if I had a heterozygote parent, so I have a dominant and a recessive crossed by a pea plant that is both recessive, some of those will be white. This guy and this guy and this guy will make white flowers. So a test cross is when you have a unknown organism with a dominant trait. You cross it with a homozygous recessive just to see what it is. We're not going to go into incomplete dominance. Basically, Mendel was lucky. If it didn't work, a lot of times that this is complete dominance. A lot of things in life aren't completely one or the other. I mean, if you look at humans, right? You're not just tall or short. There's a large gap in height, right? You can be somewhere in the middle. You can look at skin color, hair texture. You look at animals, right? Their skin tone. There's blending. You look in nature. There's lots of different colors. So this is incomplete dominance. Like I said, we're not going to really spend a lot of time in that incomplete dominance, but I just want to make you aware of it. It is not just yes or no, one or the other. This is a great summary page, though, to just make you aware of and to make you think and realize. So our big ideas for this was that we have chromosomes carry heredity or genes. Chromosomes occur in pairs. We have new combinations of genes that occur in sexual reproduction. We're only focusing on monohybrid crosses. We are not focusing on dihybrid crosses. And Mendel had three principles. He had the principle of dominance, which is one allele is louder than the other, so it covers up the other. We have the principle of segregation. So basically, genes separate from each other in gamete formation. So when we have sperm and eggs forming, the chromosomes don't stick together, they separate. We have the law or principle of independent assortment. So members of one gene pair, they separate independently. So they don't stick with their buddy. They have just as equal of a chance of separating whenever or however the gametes form. So your assignment for this is to go through the notes with the genetics as you listen to this video. I'm hoping it was relatively short. We will see when I pause and stop this. Then you will do some Punnett square practice, and you will read a little bit about Mendel, and then we will see where we go from there. If you have any questions, please feel free to email and text me or call me, and I would be happy to help however I can. Talk to you guys later. Bye-bye.